grade 12. It's me, Sir Ramon. I'm here to summarize Non-Mendelian Inheritance. Now, you guys have had readings on Khan Academy, and I'm sure you guys have taken the time to read it if you're even on Khan Academy. But these slides and this discussion is so that you guys can understand better exactly what non-Mendelian inheritance is about. So when we say non-Mendelian inheritance, it means that Mendel's laws, the law of segregation, the law of independent assortment, and the law of dominance are not necessarily followed. So first things first, what if the dominant trait, yung dominante, hindi siya totoong dominant? Again, if it's not completely dominant, what happens to heterozygous individuals? So when we say heterozygous, we do mean those individuals that have a dominant allele and a recessive allele. Wow, I can't draw. So again, a dominant and a recessive allele. Now what happens to them is that we have incomplete dominance. Again, when the dominant allele can't completely mask the recessive allele, that what happens is that the dominant trait and the recessive trait are going to blend with each other. Again, they're going to mix with each other. And so we get what we call intermediate phenotypes, or what we call blends. Again, blending of phenotypes. One example of incomplete dominance is in your flowers. Some flowers have red and white, and when they go together, again, your dominant allele, C capital R, and your recessive allele, your C capital W, when they go together, you have pink individuals. Again, red plus white equals pink. You have the blending or the mixing of your phenotypes, resulting in what we call intermediate phenotypes. Again, incomplete dominance, intermediate phenotypes. Alright? If both alleles are equally dominant, again, we don't have a dominant allele anymore. They're both dominant, the same level of dominant. Your heterozygous individuals would have multiple dominant alleles, which means that both the dominant traits are happening at the same time. And so we have what we call codominance. One example is your coloration called roan coloration. So here we have a red mumu and a white mumu. And when they go together, their offspring can be both red and white mumu. Again, it's not pink. We don't see pink cows anywhere, but we do have red spotted white cows, or roan colored cows. Again, roan is not necessarily red plus white. It's any combination of two colors that result in splotches. Alright, so again, instead of blending, we have both of the phenotypes happening at the same time. Okay? Another example is your ABO blood group. Kung ano man yung blood type ninyo, I'm blood type O. Alright? Blood type A and B, these are expressed due to the alleles of blood type A and blood type B. So again, we'll see more of that after we discuss the multiple allele system. This is when there's more than two alleles. Again, normally we have just a dominant allele and a recessive allele, the capital and the lowercase. But what if it's not just A? What if you also have allele B and it's also for the same gene? Again, for the same gene. Alright, that gives you multiple alleles. There's more than two alleles that determine a trait. And another, this is also a characteristic of your ABO blood group. So it's both codominant and has more than two alleles. So in your ABO blood group, there's three alleles: the allele for A, the allele for B, and the recessive allele I. Again, the recessive allele I, this is the one that codes for your blood type. Oh my god, it's hard to do this with the mouse pad. Blood type O. Alright, so me being blood type O, I have the genotype of small I. So my red blood cell has no antigens. There's no identifying factors, identifying proteins on the periphery of my red blood cell. That's why blood type O can donate to everybody, because there's no ID. Okay, hindi sila pwedeng hulihin kasi wala naman silang ID na nagsasabi na bawal sila dyan. Alright? Blood type AB, on the other hand, has both the capital IA and capital IB alleles, which gives them the identifiers for both A and B. Alright? So these ones are very hard to donate for. They can receive any blood type because they recognize it as their own. Again, blood type A, no problem. We also have that ID. Blood type B, we also have that ID. That's fine. Again, they can receive any blood type within the ABO blood group. But you can't just go donating blood type AB to other people, okay? Because again, it has two IDs that will allow the body to recognize them as foreign bodies, all right? Your blood type B and your blood type A can have two possible genotypes. You have homozygous dominant or heterozygous, all right? Okay, so again, blood type is both multiple allele and codominant. Moving on, if we have one gene that determines multiple characteristics, again, one gene, multiple characteristics, what happens is what we call pleiotropy. Again, pleiotropy is when one gene will determine many features. It's no longer 1:1. Again, before Mendel said one gene, one characteristic. In this case, one gene will go to many characteristics. All right. So your example of this is sickle cell disease. So sickle cell disease has many characteristics, but most apparent is that your blood cells look like sickles. Again, instead of the normal cute flattened sacs, they look like sickles, and that causes many types of characteristics to occur. They can't flow smoothly and are usually destroyed by the body. So because of that, you have lethargy, you have nausea. It has many effects on the characteristics of an individual. So again, secondary effects, you have organ failure, brain damage, lots of nasty stuff. And if you look here, lots of stuff that has similarities to COVID symptoms. Oh no. But what if it's the other way around? What if many genes determine just one trait? Again, many genes, one trait. It's the other way around. This is what we call the polygenic inheritance. Poly meaning many, and gene is genes. Okay? So if you have many genes that are relating to one trait or one characteristic, that's called polygenic inheritance. Our example of this is skin color. So in skin color, this is a very simplified way of explaining skin color. Again, it's a spectrum, because if you have all recessive alleles, for example, just for an example, if you have all recessive alleles, the non-shaded circles, okay, non-shaded circles, you'll have very fair skin. But the more dominant alleles you have, again, the more black alleles you have, the more shaded alleles you have, the more shaded your skin color becomes. And these alleles come from different genes. Alright, so again, one characteristic, which is skin color, determined by many different genes. Now we get to epistasis, which wasn't discussed that well in Khan Academy, but for our discussion, we'll just focus on the primary definition: one gene influencing how another gene is expressed. Again, one gene expresses differently because of the other gene. So this is against the law of independent assortment of Mendel, meaning hindi na sila magkahiwalay ang magkakaibang genes. They affect each other now. And they affect each other based on their genotype. So the classical dihybrid ratio, the one that I asked you guys to memorize after we had our example of dihybrid cross, is 9:3:3:1. 9 both dominant, 3, one of them is recessive, 3, another one is recessive, and 1 for both recessive. For epistatic ratios, again, one gene will affect another gene. Okay? So depending on how it is affected, you have variations of this ratio. You don't have to memorize this, but you do have to understand how to manipulate your dihybrid ratio. Again, if we say that one gene affects another, and depending on how it affects the other gene, you have to be able to modify this ratio to get the type of interaction. Again, no need to memorize the names, but the numbers and how we get those numbers, you'll have to understand. For example, in your classical ratio 9:3:3:1, what if having dominant epistasis, meaning, okay, if the dominant trait, again, if you have the presence of at least one dominant allele on either gene, it makes the other gene express as dominant as well. Again, if you have one dominant allele and it expresses so that this one will not express as A, small b, it expresses the same as this one, you have what we call dominant epistasis. Again, you just put together the things that would end up expressing in the same way. Since this will express the same as this, You have 9 plus 3, you get 12. Again, just read problems. Most likely the problems will say that this phenotype actually has this genotype. Okay? So if you have two genotypes that express the same way, you most likely have an epistatic ratio or an epistatic relationship. Alright? So you will have to manipulate your 9:3:3:1. Okay? If you have further questions about this, feel free to message me. I'm going to answer any and all of your questions. But moving on, we have sex-linked inheritance. So this isn't as fun as it sounds. It just means that your inheritance is linked to your sex chromosomes, your X and Y chromosomes. On the other hand, if we say body chromosomes, there's no such term as body chromosomes. We call them autosomal chromosomes. Alright, so you have the sex chromosomes and autosomal chromosomes. If we focus on the sex chromosomes, we have sex-linked inheritance. This means that the genes are on the sex chromosomes themselves. Okay? And it depends whether it's on the X chromosome, which makes it X-linked, or if it's on the Y chromosome, which is Y-linked. Now, most sex-linked disorders are recessive disorders, and they only most of them affect mostly males. Alright? Because for X-linked genes, males only have one X chromosome. Again, males have X and Y. Wow, I can't draw. Okay, so X and Y. So we only have one chromosome, one chance in order to have a normal set of genes. So if you inherit a gene that codes for a disorder, you have no other extra chance in order to get a normal gene. On the other hand, for the ladies, since they're XX, they have two chances to get the normal gene. Again, the normal allele of the gene, I mean. Alright, so even if they get the gene that codes for the disorder, their other X chromosome might have a copy that is normal. Alright, so that is usually a dominant allele. The normal allele is usually dominant, and it will mask the effects of the disorder allele. For Y-linked sex disorders, only males have a Y chromosome, so only males will have trouble with that. Okay? So, lugi talaga mga lalaki in terms of sex-linked disorders. But females can be carriers of X-linked recessive traits. When we say carriers, that means that normal sila. Again, they have no problems in their own personal life. However, they carry the recessive allele. They carry the allele for the disorder. So in this case, again, we have the genotypes. If they have capital A, again, both normal, they have the dominant or the normal phenotype. But if your girl Lou has one recessive allele, okay, that makes her a carrier, meaning she does not display the symptoms of the disorder, but her offspring can inherit a recessive allele. This is why a lot of times in families you have skipping generations of colorblind males, because the females usually are carriers of that recessive allele. Now, when we say sex-influenced inheritance, it just means that different sexes express the same genotype differently. One example is baldness. You can have the same genotype between a guy and a girl, but the guy would end up bald and the girl will not. That's because the difference in sex will make it express differently. Again, it can be the same genotype, for example, capital A, but for guys, capital A, small a will code for bald, but for girls, it will code for normal. Alright? Sex-limited traits are limited by the biology of the sex. So, for example, cervical cancer, okay, the cancer of the cervix, you guys should know by now that guys don't have a cervix. That's only in the female reproductive system, so only females can have cervical cancer. So it's a sex-limited trait. Now, moving on to pedigree analysis, this is basically tracking a trait through the family tree. Pedigree is not dog food, that's just a brand name of dog food, but a pedigree is basically a family tree that traces a trait throughout the family. Okay? So when we analyze a pedigree, the first thing we need to ask is it dominant or recessive? Again, is the trait dominant or recessive? So dominant traits will never skip consanguineous or directly related generations. Again, it will never skip directly generated directly related generations. Again, if your mommy and daddy give birth to offspring, that means they're consanguineous. They're very much related. Ngayon, kapag si baby lumaki at nag-marry ng someone else, they're not consanguineous. They're not directly related. Unless meron kang sketchy stuff happening and you're watching anime or something. Alright? Again, consanguineous, directly related. Connected sila by a vertical line of mommy, daddy, baby. This is basically the parental line or the ancestral line. Okay, so they are consanguineous. So if it doesn't skip, then that means that the trait is most likely dominant. But if it does skip, again, if you see it skipping a generation, so it's present in one generation, in the next generation it's gone, but then in the generation of offspring, okay, then it skipped a generation, then that means it's recessive. Again, if in the P generation it's present, in F1 generation it's absent. Oh my god. And then in the F2 generation it's present, and they're connected by lines of consanguinity. Again, they are directly related to each other, just a continuous vertical line between these three generations. Then you have what we call a recessive inheritance. That's just the first question. The second question, is it sex-linked or autosomal? So autosomal traits will affect both sexes equally. It's just a matter of counting how many guys are shaded and how many girls are shaded. Again, take note that for guys, our shape and pedigrees is the square kasi yung guys lang naman yung merong pantusok. And the girls naman, they're circular. Okay? They're circular because, no, yes. Okay, so again, Y-linked only males will exhibit them, only males, while X-linked, it's mostly males. But it doesn't end there. Just because you check the proportions doesn't mean that it is for sure. It's just the most likely. So you have to check if your guess is correct by figuring out the genotypes. You have to see if the mode of inheritance makes sense. Okay, so you guess the genotype of the lower generations and trace it back up to the parental generations. Okay? For example, in this case, we have a shaded individual here and then non-shaded here. Okay, so if we look at this, the first question, dominant or recessive. Okay, so in the parental generation, they're both white, they're both unshaded, and then suddenly someone's shaded. That's a clear-cut example of skipping. Again, this generation, no trait. Suddenly, lumitaw siya bigla. The only way that could be possible, is if it is a recessive trait. Now, the second question, is it autosomal, X-linked, or Y-linked? Now, this is a very small pedigree, so it's very difficult to make a guess whether it's autosomal or sex-linked. So we'll skip that, but let's see if we can guess the genotypes. Again, let's guess the genotypes. So we're assuming that this is recessive, right? So if we assume that that's recessive, okay, let's give it a genotype of homozygous recessive. So individual number 4 here is homozygous recessive, small a, small a. Okay. Now, what combination of genotypes can produce a homozygous recessive, but both parents are recessive? Again, if both parents have dominant phenotype. Again, for this case, since they're unshaded, we know that they have a dominant phenotype. And dominant phenotypes have capital A and either big A or small a. So let's leave it blank. Again, whether it's big or small a, we know that as long as there's one capital allele or a dominant allele, it will have a dominant phenotype. Alright? So, what cross can we have between dominant phenotypes so that we can have a recessive phenotype? Again, one allele from each parent will be given to the offspring. So this baby number 4, has to get one recessive allele from daddy and one recessive allele from mommy. Okay? So, unahin natin si daddy. Okay? Kung capital na yung isang allele niya, anong kailangan niya na allele para maibigay sa anak niya para maging recessive yan? Siyempre, kung capital A to, if that's capital A, again, if that's capital A, daddy will no longer have a recessive allele to give to baby. Okay? So, imposibleng capital or dominant allele yung blank natin for daddy. So, ang ilalagay natin dyan ay recessive allele. Your father must give a recessive allele to the offspring. But there's another allele from the mother. And it's the same case if it's capital or dominant allele here. Again, if it's capital or dominant allele here in the blank, it will not be possible to have a recessive offspring. So it has to be a small letter as well. Kasi ito naman ang ambag ni mommy. Okay? In this way, we know that these individuals, si mommy and si daddy, are heterozygous individuals, both of them. Capital A small a and capital A small a. Now for the next, for 3 and 5, we have no way of knowing if they are capital A or capital A and small a. Alright? So even in the case of 5, 6, 7, 8, it's very difficult to determine what their genotypes are. But at the very least, our guess that this kind of inheritance is recessive makes sense because we have a homozygous recessive genotype for 4 and we have heterozygous genotype for our parental generation. So it can make sense. It's possible for it to be a recessively inherited trait. Alright? So next we discuss where the genes are located. So these genes are located where? They're located on the structure that we call loci on the chromosomes. Again, loci or locus is just the specific, it's just the specific place that they are on the chromosome. So when we say genes are linked, that means that these genes are closely located. Again, their loci are very close to each other. And since they're very close to each other, they're usually inherited together. What does that mean? It means that in the chromosomal theory of inheritance, genes don't independently assort. What does that mean, sir? What the heck? Let's go back to our meiosis. Again, in meiosis, we have crossing over. That's when your chromosomes cross over and then they exchange. They exchange their genetic material, right? That happens in prophase 1. And then crossing over, we have what we call recombinant chromatids. Again, your crossing over produces recombinant chromatids. That's a complicated term for nag-mix and match sila. So in this case, okay, the mixing and matching produced recombinant chromatids. And chromatid is just one of the sister chromatids. So our recombinant chromatids are just the ones that did the exchange. The chromatids on the sides, again, the chromatids on the sides are still non-recombinant. Again, the ones on the sides are non-recombinant. Kasi, hindi naman sila nag-mix and match. Hindi sila nag-exchange or crossover. Okay, so we have recombinant in the middle and non-recombinant on the side. So take note of that. So again, we have two types of chromatids. The ones that with no crossover, your non-recombinant chromatids, are called the parental chromatids. Whereas the ones that cross over are called recombinant chromatids. And recombination is very rare. Since it's rare, your recombinant chromatids will have very small proportions, whereas your parental chromatids, sila yung mas marami. Okay? So again, recombination is very rare, and we can measure how often it occurs based on what we call the recombination frequency. But we need to do a modified test cross. Now, when we say test cross, again, in our Mendelian genetics, a test cross is when we cross something that we don't know with homozygous recessive. In this modified test cross for recombination, we have a tester that's homozygous recessive for 2 genes. Again, we have to look at 2 genes kasi in recombination, what we're looking at is the relationship between gene number 1 and gene number 2. Gaano sila kalapit? Okay? Gaano sila ka-linked? Alright? So, again, 2 genes. So when we cross them, we get phenotypes, 4 different phenotypes when we're discussing just 2 genes. And when we look at these 4 different phenotypes, we can say that 2 of them are parentals because they're the common ones. Again, yung parentals, they're the common ones kasi hindi sila nag-recombine. Hindi sila nag-crossover. The rarer ones, the rarer phenotypes, this is most definitely the recombinant phenotypes. Alright? So again, these are the phenotypes that result when there is crossing over of the chromosomes. So now that we know the number of recombinants and the total number of offspring, we can get what we call recombination frequency. How often these two genes recombine with each other. Okay? So we just get that by getting recombinance divided by total offspring. So again, we just get the few, we divide it by all the number of offspring, and we multiply it by 100% para maging percentage siya. Okay? And when we have recombination frequency, we can link it to genetic linkage. When we say genetic linkage, again, the linking between genes is how close they are to each other. Okay? So we can link recombination frequency to genetic linkage again if they are far from each other, if your genes are far from each other. Okay? If they're far from each other, there's more space between them for them to recombine. Again, that large space will allow them to have a point of crossover. Alright? What does that mean? So kung maliit lang yung space, natin, for example, in this case, very small space, that's the very small space kung saan pwede sila mag-crossover at magkaroon ng recombination between gene A and gene B. Alright? Again, the smaller the space, the harder it is to have a recombination event where magkakaiba si gene A tsaka si gene B. Nag-iba sila ng pwesto. Okay? Kasi maliit lang yung space between them. But if you have a large space between them, again, the farther the distance between genes, the higher the frequency of recombinance between them. Again, the larger the space for them to cross over at magkaiba yung positions ng dalawang genes, mas mataas ang RF mo. Mas mataas ang distance mo. And the farther the distance is, the less there is linkage between the genes. Again, the farther the genes away, the less linked they are. Okay? So this allows us to map out our chromosomes. Even without looking at the molecules or the chromosomes themselves, we can figure out what gene locuses or loci are close and which ones are far apart from each other. Okay? So note that the recombination frequency, the maximum is 50%. And the reason for that is because recombination is very, very rare. So your parentals should always be equal to or more than your number of recombinants. So hanggang 50% lang. Kasi kung sumobra yung 50%, ibig sabihin, mas marami na ang recombinants mo kaysa sa parentals. That don't make sense, bro. Because again, we have recombinant chromatids in the middle and parentals on the sides. Okay? So we can make linkage maps, which is a map of relative distances of gene loci, by comparing the frequencies. Yung malaking frequency, the largest frequencies, will be the ones that are on the outside. When we say outside, sina-sandwich nila yung dalawa pang genes. For example, in this case, the recombination frequency between A and C is the largest. So we can say that A and C are the ones on the ends of your chromosome. Whereas nasa gitna naman si letter B. Again, si gene B should be in the middle, the locus of gene B. And your recombination frequency should approximately add up to each other. Again, 13.2 plus 6.4, it's sort of close to 18.5. You'll get 19.6. So, medyo, again, hindi siya saktong science, pero medyo nag-a-add up sila. Again, we work in trios, in threes, in order to figure out where the loci are, how these genes are arranged on the chromosome. Again, yung malaki, yung recombination frequency, sila yung pinaka malayo sa isa't isa. So that has the largest distance, so that means that it's possible to have the other genes in the middle. So if you add up the recombination frequencies of the genes that you think are in the middle, you can figure out which genes are in the middle of the two outside genes. Again, we work in trios, in threes. So again, when we make linkage maps, you work in trios. Even if you're given more than 3, you choose the 3 that look like they will add up to each other. Again, you choose 3 where the big RF is approximately equal to 2 small RFs added together. So if you draw a line and you mark the loci so you don't lose track, you should be able to make a diagram similar to this so that you can make your linkage map in an orderly manner.